be presenting on a very cutting-edge technology of specific gene targeting in zebrafish genome using CRISPR-Cas9 approach.
Thank you so much. Hi guys, thank you so much for the introduction. I'm Melanie Martinez.
I'm Alina Balasa.
I'm Mitsuko Morita.
I'm Kyle Igner.
I'm Kristen Stewart.
And we are currently conducting research under Dr. Daniel Zito to analyze the specific gene transferring in the zebrafish genome using the CRISPR-Cas9 approach. So our research aims to test the effectiveness of our design scheme to disrupt two genes known as OEP and albino. These genes are respectively responsible for the phenotypes of cyclopia and albinism, which we'll be discussing later on. And these generation of zebrafish mutant lines can be used as a genetic model to help us further understand the genes required to regulate the normal biological process during development.
So we chose zebrafish as our model organism to use to induce and study human diseases. We chose zebrafish for a number of reasons. The first reason being that it's very cost effective. The second reason being that the genome has already been sequenced, which makes it a very feasible model. Just due to the fact that there's readily available genes that are used for easy targeting. The next reason that we chose zebrafish is that since the genome has already been sequenced, this makes it easy for zebrafish to be used as a standardized human biological model in order to study for future experiments. And then zebrafish actually have a very rapid development for embryos. The first 9 cell divisions are 15 minutes apart and can be seen as early as 24 hours, which makes it very easy to track the development of the embryos for our prospective candidates. Our first gene of interest is going to be OEP, which stands for one-eyed pinhead. It results in the disease called cyclopia. Cyclopia is an autosomal recessive genetic disease that is lethal and usually leads to stillbirths and miscarriages. And as you can see here, it presents as the phenotypic presentation as a lack of a nose and the 2 orbital sockets converging into one, therefore leaving the eyes to be above where the nose should be. The gene that we targeted was OEP. The consequent phenotype is cyclopia. And it's an early onset gene, meaning that you can see the apparent phenotypic presentation earlier in the embryonic development. And you can see here at the top, we have a wild type zebrafish, which just means a normal zebrafish. And then at the bottom here, you see our OEP zebrafish, which you can see that the lack of the nose leads the 2 orbital sockets to eventually be able to converge into one. Our second gene of interest is going to be the albino gene. Albinism is another autosomal recessive genetic disease that leads to malfunctioning melanocytes producing a phenotypic presentation of a lack of pigment in the hair, skin, and eyes. The gene that we targeted is albino. The consequent phenotype is going to be albinism, and it's a late onset gene, meaning that you will not be able to see the apparent phenotype until later in the embryonic development. And then here, I'm sorry. No, you're right. It's fine.
So I'm sure many of you have heard of the CRISPR-Cas9 approach, but I'll be briefly discussing exactly how it works and its application to our experimental design. So this Nobel Prize winning gene editing technology is used to alter the genetic information in a specific organism. So this system consists of 2 different components. The first being a small guide RNA sequence that directly matches the desired target genes of our model organism. In this case, that would be the OEP and albino gene that is in our model organism, the zebrafish. The second component would be the protein called Cas9 DNA nuclease that functions as molecular scissors in order to promote a double-stranded DNA break in the sequence. This will ultimately result in a loss of gene function that we can then use to further understand the requirement of that gene and for the proper development of the organism.
Okay, so with that background, I'll give a brief overview of the experimental aims of our research. First, we created small guide RNAs for each of our genes of interest, the OEP and albino gene. Next, we constructed a DNA plasmid for each of those genes of interest that contain that CRISPR-Cas9 delivery system, which includes the specifically designed small guide RNA, the Cas9 gene and the GFP gene, which stands for green fluorescent protein, which will allow us to trace our results afterwards. Next, a male and female zebrafish were bred to obtain our healthy embryos. We used a microinjection delivery technique, which included the creation of a microneedle to physically insert the DNA into the embryos. This must be done while the embryos are still at that singular cell stage to ensure that the genes are incorporated successfully into all subsequent cell divisions throughout the development. Finally, we screened for potential candidates under a fluorescent microscope 24 hours after we injected in order to find the expression of that GFP protein. With a higher expression, we can conclude that we successfully injected our embryos, as you can see there, that last picture.
Thank you. All right. So in summation, the overall goal of our research project was to be able to effectively target and disrupt the gene sequence. So the two gene sequences, OEP and albino, thus allowing for the development of their characteristic phenotypes of albinism and cyclopia. Here are a few of our isolated specimens in the lab. On the left-hand side, you can see the characteristic green fluorescent protein being illustrated here, indicated by the red arrow. And on the right-hand side, you can see another one of our isolated subjects that didn't have the uptake of the green fluorescent protein, characterized by the dark space under fluorescent screening. Go ahead and go to the next one again. All right, and then so from our research, we've been able to see that the illustration of the green fluorescent protein under the fluorescent screening illustrates the possible integration that took place between the GFP protein and the Cas9 protein into the DNA of the biological model of the zebrafish embryo. Future experiments and future applications of our research we'd like to apply to would be the determination of gene specificity of gene disturbances and disruptions of the sequence, along with establishment of a permanent zebrafish mutation line, so that we can further analyze the DNA and the sequencing and characteristics. And then
And we would just like to take a second to thank CBU and the Natural and Mathematical Research Symposium for having us today. We also would like to acknowledge Dr. Daniel Zito for having us participate in your research. It's been an irreplaceable experience that I know we're all very grateful for.